Okay, guys, we will make a start now. So welcome everyone to our biology session. And in today's lesson, we are going to be looking at enzymes. Okay, we're going to start off by looking at an overview of enzymes and the activation energy. We're then going to move on to the induced fit model of the enzyme. And then we're going to finish off by looking at the rate of enzyme reactions. Okay. So this is what the specification states. We're going to be covering all the points on the spec in today's lesson. Okay, and we're going to start off by looking at the activation energy. Okay, so first of all, what is the activation energy? Okay, and this is the definition of the activation energy. The activation energy is basically the minimum amount of energy um, required for a reaction to occur. Okay, and basically, if we think about this, yeah, you would have looked at this in chemistry, but if we had a look at this reaction, this is for an endothermic reaction. Okay, this is the reactants, and this is the products. Okay, and this much energy is required in order for the reaction to take place. Okay, we've got energy on the y-axis. That much energy is required for this reaction to take place. Now, what a catalyst does, or what an enzyme does, it reduces the amount of energy required for the reaction to take place. So it lowers the activation energy. So instead of this much energy being required, it can lower it to this much. So only this much energy is required in theory. So what it does, it is that it lowers the activation energy. Okay, so activation energy over here, we saw that a lot of energy was initially required, and then after we added the catalyst, much less energy was required. It lowers the amount of energy required for the reaction to take place. Okay, and that's what basically the activation energy is. When an enzyme attaches to the substrate, they can lower the activation energy needed for the reaction to occur, therefore speed up the reaction itself. So imagine this cycle over here. We've got this. If we have less energy requiring for the reaction to take place, like over here, we have less energy taking place. Um, what actually happens is that because less energy is required, the reaction can occur faster. Over here, because more energy is required, the reaction will occur slower. So that's another benefit of having a lower activation energy, is that less energy is required, so the reaction can take place faster, which means more reactions can take place in a smaller amount of time. Okay, so before we actually dive into enzymes, it's really important that you understand what activation energy is. And it's a concept you would have looked at GCSE, but we're just recapping here at A level. So the main things that you need to take away from this slide is that the purpose of the activation energy is the amount of energy required for a reaction to take place. And enzymes act as catalyst. Okay, they catalyze the reaction. And because they're catalyzing the reaction, what they're actually doing is that they are lowering the activation energy. If they're lowering the activation energy, that means less energy is required to turn the reactants into the products. And if less energy is required, that must mean more the reaction can take place faster and more reactions of the same type can occur in a shorter period of time. Okay, so that's basically the activation energy. Now, this is the lock and key model. Now, you would have looked at the lock and key model at GCSE, and it's not really touched upon at A level. However, you must be, there's a new model of enzymes that you are going to learn in this lesson. Questions could ask you to compare the two models. Okay, so just to recap on the lock and key theory. Okay, so as you would have known from GCSE, the enzyme is the lock and the substrate is the key. Okay, so if I was to draw a diagram, we could have this and we could have this. This is our enzyme and this is our substrate. Okay, and as you know, what's going to happen is that this part is complementary, which means has the same shape as this part from the substrate. And what's going to happen is that the substrate is going to bind to the enzyme's active site. So if we just label this, I'm just going to switch colors. Okay, so we know that this is our enzyme. We know that this is our active site. We know that this is our substrate, and we know that the substrate is going in and binding to the active site of the enzyme. Okay, and the reason why, it's because it's complementary. That's the term that we use, complementary. It's complementary in shape to the active site from the enzyme. Okay, so that's why we stated the enzyme is the lock and the substrate is the key. And the substrate is going to bind to the enzyme on the complementary active site. Okay. So the substrate fits like a key due to the complementary shape of the active site, which we've already said. The enzyme's active site has a fixed shape. Now, I've put this in bold. The reason why is because this is a key difference to the other model that we are going to be looking at. The enzyme's active site has a fixed shape. Okay, what does that mean? This means that the enzyme's active site is not going to change shape. So if I was to draw another model out of an enzyme, let's pretend this was an enzyme, and you can see that this is the active site of the enzyme. It's going to be permanent. It's not going to change shape. Okay, there's only one active site. It's not going to change shape. What's going to happen is that the substrate is just going to bind into the active site of the enzyme. That's it. Okay, and the substrate is going to collide with the active site of the enzyme, and then it's going to bind to the enzyme. And this is a new term that we are going to be using every single time. And on mark schemes, this will always be worth a mark. So just learn this term, and whenever you see an enzyme question, just put in enzyme substrate complex into the answer, and you are basically putting your bets on that you will get at least 1 mark in that question. Okay, this forms an enzyme substrate complex. So this is the new part. Okay, so when we've got the substrate binding to the enzyme, what we will have is we will have something like this. So we would have the enzyme, and then we would have the substrate bound like this. Okay, can you see it's bound over here? Now, that's the name of this structure. This structure is called an enzyme substrate complex. That's what it's called. An enzyme substrate complex. That's the name of the complex that is formed. Whenever something binds to another thing, you form a complex. Okay, and you will realize this when you study topics further on. Yeah, there's for example, when an antigen binds to an antibody, you form an antigen antibody complex. There are many complex that that are formed, and they're relatively easy to remember. Over here, you've got an enzyme binding with a substrate. That's why the complex structure that is formed is basically the two things, enzyme and substrate, combined together with a complex at the end. That's why it's called an enzyme substrate complex. Just learn that phrase. So once the enzyme substrate complex is formed, the substrate slightly distorts in shape, and this distortion can lower the activation energy. So what's actually happening is once we form this, the active site from the enzyme is going to put in some energy to break the substrate into pieces. Okay, so what's going to happen um, is that it's going to distort the shape of this substrate. And you're going to eventually, I'm running out of space, I'll draw it down here. So we originally had this, okay? We had the active site of the enzyme being bound to the substrate. Now the enzyme is going to break that substrate down into individual pieces. It's going to break it down into two smaller pieces. And the reason why is because it distorts the structure, which means it breaks the structure of this substrate. Because it's bound here, it's just going to literally cut the substrate in half, and you're going to form two separate, smaller products. Okay, and that's basically the lock and key model of the enzyme. It's nothing new. It's exact same from GCSE. The enzyme is the lock and the substrate is the key. Think of it that way. The substrate will bind to the enzyme at the complementary active site. The enzyme's active site has a fixed shape. It's not going to change shape. And once the substrate is bound to the active site of the enzyme, that's what you call an enzyme substrate complex. Okay. Now, once it bound, the enzyme will distort the substrate, and then that will cause the substrate to break into its products. Okay, and that's basically the lock and key model for you. Okay, now the new model that we have now, because this model is slightly outdated, so we now have a new model that has been introduced into the A-level biology spec that you are required to know. And this is now the accepted model of the enzyme. Just a minute, guys. Okay, sorry about that, guys. Um, So we were talking about the induced fit model. There is a new model which is now accepted. It's the newer model because the lock and key model is slightly outdated. Okay, and that's the induced fit model. And this is the detail that you need to know for this model. So the enzyme is like a glove and the substrate is like the hand. Okay, think of it that way. So in this model, we saw that the enzyme was like the lock and the substrate was like the key. Um, That's why it's called the lock and key model. This is called the induced fit model, or you could have said the glove and hand model if you wanted to. But just your induced fit model. The glove and hand is a nice analogy of thinking about what actually happens. So this time the enzyme is like the glove and the substrate is like the hand. Now, the glove is not exactly complementary to the shape of your hand, but when you put your hand into it, the glove actually molds around your hand and becomes completely complementary to your hand. Obviously, yeah, there are many different size hands. We all have different size hands, but we wear the same gloves. Okay. Normally you only get three sizes of gloves: small, medium, and large. And they normally fit whoever wears them. Anyway, and the reason why is because the glove is like the enzyme. Our hand is the substrate. We are putting it into the glove, um, and initially it's not complementary. However, the glove will change shape, it will move, it will become larger or smaller, and it will sort of wrap around your hand and it will become complementary to your hand. And that's basically the analogy to remember the induced fit model. The induced fit model is when the active site of the enzyme is induced. Induced means it changes shape, okay? Or slightly changes shape. I've got it down there. Slightly changes shape to mold around the substrate. So if I was to draw this out, I'll draw it out over here. Let's say we've got this enzyme, and um, we've got this massive substrate. Okay, now it's not perfect, but as you can see, this is not going to be a perfect fit into this active site of the enzyme. So what's going to happen is that this enzyme is going to come to this substrate and it's going to wrap around this substrate. It's going to change shape ever so slightly, and it's going to wrap around this shape. So it's going to wrap around the shape. So you're going to have something like this. I know it's not exactly in shape, but just to get an idea, we've just changed the shape ever so slightly of the active site. And the active site shape has slightly changed, and now it is complementary in shape to the substrate, and therefore it can bind to the substrate. Okay, so whenever it's bound, yeah, this complex is always still going to be called an enzyme substrate complex. So enzyme substrate complex. Okay, and when the enzyme substrate complex forms, the enzyme, as you can see, has become a bit larger and it's molded around the substrate. It's slightly changed shape um, uh, to fit the substrate. This is a phrase that you need to remember. It puts strain on the bonds. Okay, it puts strain on the bonds. And mock schemes will have that, or it puts stress on the bonds. Or something like that, strain, stress on the bonds, and that causes the bonds to weaken. And when the bonds weaken, it lowers the activation energy, and therefore the products can be released, and then the active site can return back to its original shape. Okay, so we had an enzyme, we had a substrate over here. So we have our enzyme and we have our substrate. What's going to happen is the substrate, the enzyme is not the active site. This part of the enzyme is not fully complementary to the active site of this active site of the enzyme is not fully complementary to the substrate. Okay, so it's going to struggle binding. However, what's going to happen is that the enzyme is going to slightly change shape. The active site of the enzyme is going to slightly change shape in order to fit the substrate. And that's why it's done here. It's slightly changed shape. And because it's slightly changed shape, it can form the enzyme substrate complex. Okay, and once this enzyme substrate complex can be formed, what happens is that strain is put on the bonds. So remember, the substrate has bonds within it. Yet we're putting stress on those bonds. We're putting strain on those bonds, causing those bonds to break. Okay, so the active site puts strain on those bonds, causing the bonds to break. And because the bonds are broken, the products are released, and then the active site can return back to its original shape. Okay, and that's basically the induced fit model of the enzyme. Okay, so far we've looked at the activation energy, we've looked at the lock and key model, and we've looked at the induced fit model. Okay, now what we're going to do is we're going to zoom in onto the structure of an enzyme. Okay, so in our last lesson we looked at proteins, and we saw that all proteins have a primary structure, secondary structure, and a tertiary structure. Okay, and then a quaternary structure is one where there are more than one polypeptide chain. Now, enzymes are tertiary structure proteins um, that can catalyze reactions. Remember, what is the tertiary structure? Well, the tertiary structure is basically the secondary structure, which has the alpha helix or the beta pleated sheet, which is processed even further into its own unique 3D shape. And then that unique 3D shape has specific ionic and hydrogen bonds present between the R groups. And because of the unique 3D shape, it can carry out its function. So for example, each enzyme has a different active site. You might have an enzyme like this, you might have an enzyme like this, you might have an enzyme like this. All of these are enzymes, but they have different active sites. Okay, the point in the middle over here is different. They have a different active site. Okay, which means they will fit different substrates. Okay, and the reasons why they have different active sites is because all of these enzymes, they have different tertiary structures. Okay, and why do they have different tertiary structures? Well, it's because they have a different sequence of amino acids. They have a different primary structure, therefore it's going to build up into a different tertiary structure. So due to different sequence So a different sequence of amino acids. So enzymes are an example of a protein. They have a tertiary structure and they have a unique 3D shape, which is able to catalyze reactions of specific substrates. Okay, so the active site of the enzyme attaches itself onto the substrate to catalyze a reaction. We already know that. And this forms an enzyme substrate complex. Again, in bold, just remember this phrase. And whenever a question on enzymes comes up, try to fit it in somewhere because it will always be given a mark. So an enzyme substrate complex is formed once the active site from an enzyme binds to the substrate. They can be used to catalyze metabolic reactions. We will come on to that later on in biology. Maybe in respiration, there's one of the processes where enzymes are used for this purpose. Okay, so that's basically the structure of an enzyme. My point over here is that we looked at proteins in our last lesson, and we looked at the four structures of a protein: primary, secondary, tertiary, and quaternary structures. And enzymes are tertiary structure proteins. And the reasons why enzymes are really important is because they all have a unique 3D shape. And that's because of the specific sequence or primary structure of amino acids that they are built up of. Each enzyme is specific to each substrate. One enzyme can't catalyze a reaction for a different substrate. It's really specific. And the reason why is because it has a specific primary structure. It will bind to the substrate to catalyze the reaction. Okay, and now going into a bit more depth on the unique active site. Okay, so the active site is specific. We already know that, and has a unique shape due to the specific folded and bonding in the tertiary structure of the protein. So remember, in the tertiary structure of the protein, we have hydrogen and ionic bonding. Now, we don't know where these bonds are going to be formed. Okay, it all depends on the specific primary structure, depends on the specific sequence of amino acids. Okay, and we don't know if it's going to be an alpha helix or a beta pleated sheet. We don't know the folding. It could fold like this, for example, like this, completely random, or it could fold something like this. Okay, we don't know the shape of the enzyme. These are two different scribbles over here. You don't know the exact folding nature of the enzyme. So number one, they're going to be folded differently. They're going to have a different secondary structure. They're going to be processed differently. You're going to have bonds formed in different places, and you're going to have a different sequence of amino acids. And because of all these factors, what you would realize is that the active site of the enzyme is really, really specific. It's really specific. The reason why is because I've already stated it enough times. Just make sure you remember that it's really specific and it can only catalyze specific reactions because it can only bind to specific substrates. Okay, the primary structure produces the sequence of amino acids in a polypeptide chain, which is different for different proteins. Okay, again, amino acids, when they come together to form the primary structure with peptide bonds, you don't know which amino acids are there. It could be a completely different sequence of amino acids. It could be similar sequences of amino acids, but you're going to have different structures of amino acids. There are a total of 20 possible amino acids that you could have, and you can't determine the order of all of them. So each enzyme will have its own specific unique active site. Okay, and as far as we're concerned with enzymes, the basics are basically done now. So we've covered the activation energy, lock and key model, induced fit model, the structure of an enzyme, and the uniquity of the active site. Now what we're going to move on to is basically the factors which affect the enzyme controlled reactions. Okay, and there are a couple of factors that you need to be aware of. Okay, so basically what this topic is what affects the rate of an enzyme reaction. So let's say An enzyme's active site is binding with the substrate. What's going to affect how fast the reaction happens? Are there any external factors which contribute to the effectiveness of the reaction? And there are 5 points which you need to know. Okay, and I'll put them here. So temperature, pH, substrate concentration, enzyme concentration, and inhibitors. These are the 5 points that you need to be familiar with, and these are the 5 points that will affect the rate of an enzyme-controlled reaction. So we've got temperature, pH, substrate concentration, enzyme concentration, and inhibitors. Okay, and we're going to take each of these step by step. We're not going to be looking at exam questions in today's lesson. Someone's asked. The only reason being is that enzymes is a really big topic. So what we're going to do today is we're just going to get through all the content of enzymes. And then in our next lesson, we're going to have a look at a required practical of enzymes. And then we're going to move on to some exam questions in our next lesson. And then that's topic 1, biological molecules, all done and dusted. Then we can move on to nucleic acids. Okay, so these 5 points are really important. Temperature, pH, substrate concentration, enzyme concentration, and inhibitors. These will affect the rate of enzyme control reactions. I'm going to start off by looking at temperature. Most of this you would have looked at GCSE, but we're still going to recap it from scratch. If the temperature is too low, what you're going to realize is that there is not enough kinetic energy for successful collisions between the enzyme and the substrate. Okay, so this is a point you would have covered at GCSE, so I'm not going to go into a lot of depth, but basically think of it from a chemistry point of view. If you have particles and you don't put in any energy, the particles are just going to stay still. They're not going to collide, they're not going to have any successful collisions. However, if you heat the particles and give them some energy, they're going to be really active, they're going to be combining with each other, and they're going to have a lot of successful collisions. So basically over here, think of the enzyme and the substrate as particles. The substrate needs to fit into the enzyme. If there's not enough, if the temperature is too low, then there's not going to be enough kinetic energy, which means the enzyme and the substrate are going to be they're going to struggle to move, and therefore, if they're struggling to move, less enzyme substrate complexes can be formed because there are less successful collisions between the enzyme and the substrate. Okay, because they're struggling to move. We've not, the temperature is too low, they don't have enough energy. Okay. Again, this is a point that you would have covered at GCSE. If the temperature is too high, remember enzymes are a protein that have bonds present between them. They've got ionic and hydrogen bonds, and sometimes disulfide bonds present within them. If the temperature is too high, what's going to happen is the enzyme is going to denature. And if the enzyme denatures, the active site is going to change shape. And the reason why is because there are bonds, hydrogen and ionic bonds, which are holding the amino acids, they are holding the tertiary structure in place. And because we've increased the temperature and the temperature is too high, it's going to interfere with these bonds. And these bonds are going to break, and therefore the tertiary structure breaks, and therefore the enzyme substrate complexes cannot form. Okay, and if the enzyme isn't able to catalyze the reaction, it isn't able to form the sort of enzyme substrate complexes, then it has no use, and therefore the enzyme denatures. So if the temperature is too low, not enough kinetic energy for successful collisions between the enzyme and the substrate. However, if it is too high, then the enzyme denatures because the high temperature will interfere with the bonds which are holding the tertiary structure in place, causing them to break, and therefore the enzyme substrate complexes cannot form, and therefore the enzyme denatures. Okay, so pretty much GCSE. If we move on to pH, this is again very similar to GCSE. If the pH is too high, there are too many hydroxide ions. Okay, so if we look at the pH scale, if we have a pH of 1 all the way to a pH of, let's go to 14, on this pH scale, this goes in increasing hydroxide ions, and this goes in increasing hydrogen ions. So increasing H plus ions and increasing hydroxide ions. Okay, so if the pH is too high, if it's like pH 14 or something, there are too many hydroxide ions. And if the pH is too low, there's too many hydrogen ions. Okay, now remember hydroxide ions are negatively charged and hydrogen ions are positively charged. If we have too many of hydroxide ions and if we have too many of hydrogen ions, what's going to happen is that the overall charge of the amino acids are going to be affected. Okay, if the pH is too low or too high, the charges of the amino acids in the active sites will it will interfere with those sort of charges. And what happens is that the bonds will break. Okay, because we've interfered with the charges of the amino acids. Remember, amino acids are joined together by peptide bonds, and these bonds, along with the ones in the tertiary structure, hydrogen and ionic, will break because we are interfering with the charges of the amino acids, which means that each of the amino acids will no longer be attracted to each other, and therefore the bonds will break, and therefore there will no longer be a unique active site, or some of the bonds in the active site will change, and therefore the active site is no longer able to catalyze the reaction it was originally catalyzing. Okay, so bottom line over here is that pH, it interferes with the charges. Okay, and the reason why is a pH which is too high means that there are too many hydroxide ions, which means a negative charge. Um, and if it's too low, there's too many hydrogen ions, which means a positive charge. And because we're interfering with the charges being too positive or too negative, what's going to happen is that the amino acids in the active site, the bonds which are holding them in place and the tertiary structure in place, they're going to break. They're going to break, and therefore the active site will no longer have a unique shape. And if it no longer has a unique shape, it will no longer have a unique tertiary structure. Therefore, it can no longer bind with the substrate. And if it can no longer bind with the substrate, then enzyme substrate complexes can no longer be formed. And therefore, eventually, if this happens at an extreme pH, the enzyme will denature. Okay. Moving on to enzyme and substrate concentration. So this is the third point on the list. Um, it's basically not that hard as well. If there is insufficient substrate, then the reaction will be slower, as there will be fewer collisions between the enzyme and the substrate, fewer enzyme substrate complexes will form. So think about it like this. Imagine we have lots of enzymes. I'm just going to draw 4 enzymes. Okay, and we only have 2 substrates. Okay, what's going to happen here? Okay, this substrate is going to bind with this enzyme, and this substrate is going to bind with this enzyme. You've still got 2 left over. Okay, the limiting factor over here is the amount of substrate which is available. Okay, so over here only 2 enzyme substrate complexes can be formed because there are 2 substrates and 4 enzymes. Yeah, 2 will bind with 2 enzymes. Okay, so the reaction is slower because there is not enough substrate present. Okay, and therefore fewer collisions between enzyme and substrate, therefore fewer enzyme substrate complexes will form. Okay, that's if there is insufficient substrate available. Okay, on the other hand, if there is insufficient enzymes available, then all the active sites will become saturated. This is when all the enzymes active sites are occupied with the substrate and they are unable to work any faster. So think about this. We have one enzyme and we have 4 substrates. Okay, this substrate will bind with the active site of this enzyme. However, what will these three substrates do? They will have to wait. Okay, that slows down the reaction. That slows down the rate of reaction because there are not enough enzymes. Okay, because there are not enough enzymes, the term that you use is saturated. Learn this term as well. It's a really important term. If all the enzymes that are working are bound to a substrate, then that means all the active sites are saturated. That means all the enzymes active sites are being used up. There are none which are available. Therefore, if you're adding any substrate, nothing's going to happen because all of them are already occupied. Just like over here, we had one enzyme, four substrates. One substrate is going to bind with the active site. The rest of the three will have to wait, and the that lowers the rate of reaction. Okay. Now, if you had an indefinite supply of both, you will get a graph which is directly proportional. So if you had infinite enzymes and infinite substrate, you will get a graph basically. That's a really bad graph. Okay. You will get a graph, still a really bad graph. Okay. Okay. You will get a graph that's basically like this. You will have enzyme concentration and you would have substrate concentration. And because there's an indefinite supply of both, that means it will be directly proportional. Okay? Because there's nothing being limiting in that scenario. Okay, so enzyme and substrate concentration. Again, fairly easy, not that much thinking. You just need to know that it exists. That's the part where people go wrong with these sorts of questions. Don't forget about these sort of easy sort of points. You've got temperature, you've got pH, you've got enzyme concentration, substrate concentration, and then you have inhibitors. And there are two types of inhibitors that we are going to be looking at. Okay, first of all, what is an inhibitor? Well, an inhibitor means that it inhibits something, it stops something. Okay, so first of all, the two categories of inhibitors. Inhibitors can be competitive or non-competitive. Okay, and this will make more sense when we look at this on the next few slides. Okay, and an inhibitor binds to somewhere on the enzyme and prevents the enzyme from functioning. Okay, so it inhibits the enzyme from functioning. It stops the enzyme from functioning. Okay, and we're going to start off by looking at competitive inhibitors. Now, competitive means that it wants some competition. Okay, the molecule is very similar to the substrate or the exact same. Okay, so if I was to draw, we have this enzyme and this is the substrate and this is the inhibitor. So we've got the substrate and we've got the inhibitor. As you know, both of these look very similar in shape. In fact, they might be the same shape. So what's going to happen is that this inhibitor is going to compete with the substrate to bind to the active site of the enzyme. So instead of the substrate binding, the inhibitor is binding to the active site of the enzyme. And because the inhibitor is binding to the active site of the enzyme, it will occupy the active site of the enzyme, which means the substrate can no longer bind to the active site of the enzyme, which means that a reaction cannot be catalyzed. Okay. Again, the name of the complex which is formed is called the enzyme inhibitor complex because you've got an enzyme, which is over here, and you've got an inhibitor, which are binding together. Enzyme inhibitor complex. So you're not going to form an enzyme substrate complex because the substrate isn't able to bind to the enzyme because the inhibitor is competing with the substrate to bind instead of the substrate. It has a very similar shape to the substrate and it's going to bind to the active site of the enzyme. Instead of the substrate, which means the enzyme substrate complex cannot be formed, but an enzyme inhibitor complex can be formed. And this will slow down the rate of reaction because the substrate cannot bind to the enzyme, which means the products cannot be released. However, if you add in more substrate, this will flood out the inhibitor, knocking them out of the active site, and the reaction can now occur. Okay, so I think this is a bit of common sense. So if you've got an inhibitor that has a similar shape to the substrate bound to the active site of an enzyme, if you just add in more substrate, this substrate is going to compete with the inhibitor as well because it has a similar shape. And what's going to happen is that the substrate is just going to knock out the inhibitor from the active site and it's going to bind to the active site of the enzyme instead of the inhibitor. So think of competitive inhibitors as a competition. You've got a competitive inhibitor which has a similar shape to the substrate and it's going to be competing to bind to the active site of the enzyme. And if it is bound, you can't form the enzyme substrate complexes. You can only form the enzyme inhibitor complexes. However, if that does happen, you can increase the concentration of substrate. So when you add in the substrate will knock out the inhibitor which is bound and it will bind to the enzyme's active site instead. And therefore, the reaction can now occur. Okay, and finally, a non-competitive inhibitor. Okay, now looking at the name non-competitive, this is not going to compete with the substrate. In fact, these are the ones which can be fatal. Okay, these bind the enzyme away from the active site. This is known as the allosteric site. Now, if this binds to the enzyme away from the active site, the tertiary structure of the enzyme will change shape, and therefore the active site of the enzyme will also change shape. And if the active site of the enzyme changes shape, the substrate can no longer bind to the active site regardless of how much substrate is added in. Even if you flood the enzyme with substrate, nothing's going to happen because the tertiary structure of the enzyme has changed and therefore it is no longer complementary to the substrate. Okay, and if I was to model this, imagine we had an enzyme over here. We have an enzyme like this, and we had a substrate like this. A competitive inhibitor will now come in and it will bind with the enzyme like this. This is the competitive inhibitor, and because that has happened, what's going to happen is that the shape of the active site is going to change shape, because the um, non-competitive inhibitor has bound, and therefore this is not able to bind with the active site of the enzyme. The substrate isn't able to bind with the active site of the enzyme. Okay, so a competitive inhibitor is going to compete. It's going to compete with the substrate to bind with the active site and form an enzyme inhibitor complex instead of an enzyme substrate complex. However, that can be resolved. How can it be resolved? But it can be resolved by adding in a high concentration of substrate, so the substrate can knock out the inhibitor. However, non-competitive inhibitor, it will change the tertiary structure of the enzyme because it will bind at a site away from the active site. And if it binds to a site away from the active site, it's going to change the tertiary structure of the enzyme, therefore the active site of the enzyme, and therefore the substrate is no longer going to be able to bind to the active site of the enzyme, which means this enzyme is useless now. It can no longer bind to that substrate because it's no longer complementary in shape. Okay, and that's all the 5 points covered of the factors which affect the rate of an enzyme controlled reaction.